Welcome. Let us discuss about hypersensitivity. Hypersensitivity. Hypersensitivity is a violent reaction of the immune system leading to severe symptoms and even death in a sensitized animal when it is re-exposed to the same allergen for the second time. Hypersensitivity is the allergy. The factors causing allergy are the allergens. They are drugs like penicillin, aspirin, airborne particles like pollen grains, spores, scales, and feathers of animals, foodstuffs like shellfish, brinjal, strawberry, infectious organisms like bacteria, virus, fungi, blood transfusion of mismatched blood. Types of hypersensitivity. It is classified in two ways. They are based on the time taken for the reaction, based on the different mechanism of pathogenesis. Based on the time taken for the reaction, immediate hypersensitivity. This immune reaction appears and disappears rapidly. Inflammatory response occurs in a few minutes. It involves the interaction of antigen and antibody. It is handled by B cells by the production of antibodies. It is antibody-mediated immunity. Delayed hypersensitivity. This immune reaction appears slowly and lasts longer. Inflammatory response occurs only after 24 to 48 hours. It involves the interaction of antigen and T cells. It is handled by T cells. It is cell-mediated immunity. Based on the different mechanisms of pathogenesis, Coombs and Gell in 1963 proposed 5 types of hypersensitivity. Type 1. Anaphylactic hypersensitivity. Type 2. Antibody-dependent cytotoxic hypersensitivity. Type 3. Immune complex-mediated hypersensitivity. Type 4. Cell-mediated hypersensitivity. Type 5. Stimulatory hypersensitivity. Of these 4 types, type 4 alone is delayed hypersensitivity and the other types are immediate hypersensitivity. Type 1. Anaphylactic hypersensitivity. Anaphylaxis. Phase 1. Sensitization. Antigen here refers to the dust or pollen in this case. The antigen enters the body through two main routes, namely the respiratory membrane of lungs and the skin. At both these places, we have specialized cells called antigen-presenting cells. In lungs, we have pulmonary antigen-presenting cell. In skin, we have Langerhans cells. These cells capture the minute antigens like pollen or dust and they take the antigens to the T cells. To be exact, the naive CD4 T cells. These naive CD4 T cells, when exposed to these antigens, become mature and are now known as mature CD4 T cells. These T cells produce a lot of chemicals like interleukin 4, interleukin 5, interleukin 13 and many more. B cells produce antibodies. The class of antibodies like IgG, IgE, IgA, IgM and IgD. Interleukin 4 acts on B lymphocytes. This leads to a phenomenon called class switching. B cells undergo antibody class switching, and so the B cell switches from making IgM antibodies to making Ig antibodies. So, the B cells which are now exposed to interleukin 4 produce Ig antibodies. The main type of antibody that is involved in type 1 reaction is Ig only. Ig antibody that is produced can identify the allergen. This antibody gets attached to the mast cells and basophils. Mast cells and basophils are the important cells involved in type 1 reaction. Mast cells and basophils have a special type of receptor on their surface. This is known as Fc epsilon R1 receptor. Antibodies have Fc part where they bind to the cells and Fab part where they bind with the antigens. Fc epsilon R1 receptor on mast cells and basophils have high affinity for the Fc portion of Ig antibodies. The basophils and the mast cells are now armed with these antibodies. These Ig antibodies are now waiting for the same allergen to enter the body and start the phase of reaction. Phase 2. Reaction. The reaction phase is subdivided into two main phases. 1. Immediate reaction phase. 2. Late reaction phase. Immediate reaction phase. It develops in minutes after the repeated exposure to allergen. Here, the preformed chemical mediators are released from the cells. As soon as the antigen re-enters the body, it is recognized by Ig antibodies that are bound to mast cells and basophils, which send signal to the mast cells to release the preformed chemical mediators like histamine, serotonin, heparin, proteases like tryptase, and chymase, ATP, and lysosomal enzymes. The most important of them is histamine. Histamine acts on blood vessels and leads to dilation. This leads to delivery of more inflammatory cells and chemicals to the site of allergy. Histamine also causes increased permeability of the blood vessels, which leads to increased transportation of cells from blood vessels into the tissue spaces. This leads to local edema and erythema. Edema is the swelling caused by too much fluid trapped in the body tissues. Erythema, which means the redness and raised temperature of the allergy site. Histamine also depolarizes nerve endings. This leads to itching and pain. Example, mosquito bite. The bump and the redness, which develops in seconds, are nothing but the immediate reaction phase. Delayed reaction phase. It develops in minutes to hours after the exposure to allergen. It involves the synthesis of new chemical mediators in mast cells. The membranes of the mast cells have phospholipids. The enzyme phospholipase converts the phospholipids to arachidonic acid. Arachidonic acid is a parent compound from which numerous prostaglandins and leukotrienes are synthesized. Leukotriene C4, D4, and E4 and prostaglandin D2, all these chemicals leads to vasodilation, increased vascular permeability, smooth muscle spasm, and edema. Type 2. Antibody-dependent cytotoxic hypersensitivity. Here, the antibodies recognize the antigens that are present on the surface of the cells or in spaces between the cells and attack these cells and lead to antibody-mediated destruction and dysfunction of the cells by various mechanisms. These antigens can be intrinsic, which means antigens which are the part of the cell membrane, or extrinsic, which means the antigens which are introduced in the body from outside. Intrinsic antigens can be anything like ion channels, hormone receptors, and nuclear antigens. In autoimmunity, the B cells produce these autoreactive antibodies which attack our own body cells and lead to various diseases. The antibody present in type 2 reaction is IgG. IgG antibodies usually circulates in the blood. Intrinsic antigens, protein antigens on the cell membranes. Example, Rh antigen on the red blood cells and GP2B3A protein present in the membrane of platelets. Antibodies against these protein antigens destroy RBCs and platelets and lead to autoimmune anemia and thrombocytopenia. Antigens present in the matrix between the cells. Matrix is like the human glue which is present in the spaces between the cells. It has numerous functions like cellular adhesion, nutrition, and waste disposal. Now, there are various diseases in which the antigens present in the matrix get involved. Example: Goodpasture syndrome. Here, the antigens present in the basement membrane of glomeruli in the kidney and alveoli in the lungs get involved. This leads to nephritis in the kidney and hemorrhage in the lungs. Pemphigus vulgaris, which is a blistering disease of the skin in which the proteins join the cells of epidermis together, are attacked by the antibodies. Receptor antigens of the cells. Antigens present on the receptors of the cells. These receptors are mainly hormone receptors, and their dysfunction leads to a lot of defects depending upon the hormone or neurotransmitter involved. Example: myasthenia gravis. Acetylcholine receptors are affected by the antibodies. These antibodies attack the acetylcholine receptors, which leads to defect in conduction of the nerves, causing muscle weakness and paralysis. Extrinsic antigens. Blood transfusion reactions. When a person is transfused with the wrong type of blood group, the antigen present on the transfused blood are recognized by the antibodies present in the blood of the person. These antibodies readily destroy the transfused red blood cells by releasing a lot of chemicals from the damaged RBCs. This leads to blood transfusion reactions. Drug reactions. Many drugs, when metabolized in the liver, leads to the formation of drug metabolites. In many cases, these metabolites deposit on the surface of the cells, which are then recognized by antibodies and attacked subsequently. The attack of the antibodies also leads to damage to the cells on which the antigens are deposited. Mechanism of antibody-mediated cell death and dysfunction. Opsonization and phagocytosis. A cell in a body which has antigens present on the cell membrane. Suppose if the antibodies are developed against these antigens, they attack the cells as they recognize these antigens. This leads to cell coated with antibodies known as opsonized cell. This process is known as opsonization. The cells which are coated by the antibodies are easily recognized by the phagocytes like macrophages and neutrophils. These phagocytes recognize those cells and phagocytose them, leading to cell death. This process is known as phagocytosis. Complement activation. The attachment of IgG antibodies to the antigens can also activate the complement pathway of our immune system. This complement pathway generates products like C5a, C3b, and C5b6789, which leads to inflammation and tissue injury. Antibody-dependent cellular cytotoxicity, ADCC. As the name itself indicates, the dysfunction of the cells caused directly by the antibodies. Example: myasthenia gravis. Type 3. Immune complex mediated hypersensitivity. The antibodies involved in this reaction belong to the class IgG or IgM. When antibodies react with antigen, they form complexes of varying sizes which are known as immune complexes. Example: systemic lupus erythematosus, SLE. It is an autoimmune disorder. In SLE, our immune system develops sensitivity against our own antigens, that is self-antigens. Normally, these antigens are present in the nucleus of the cell. Whenever a person suffers from any injury like physical injury or radiation injury in the form of UV radiation exposure in sunlight, it leads to the damage to the walls and membranes of the cells, which leads to escape of these antigens from the nucleus of the cell out into the circulation. The antibodies react with the antigens, causing the development of antigen antibody complexes or immune complexes. This is called phase of immune complex formation. Then these immune complexes get deposited in various tissues of the body like skin, small blood vessels, glomeruli in the kidney, synovial membrane of the joints, and tissues like pleura and pericardium. The immune complexes get easily trapped in various tissues because of two important reasons like very small blood vessels in the tissues and high filtration pressure. This is called phase of immune complex deposition. The immune complexes activate the complement system. C3, a component of the complement system, causes the release of histamine from the mast cells. The histamine increases the vascular permeability and more inflammatory cells, mostly neutrophils, can now cross the blood vessels into the tissue spaces. C5, a component of the complement system, acts as a chemoattractant and attracts more and more neutrophils into the tissue spaces. The neutrophils recognize these immune complexes and they try to phagocytose them. But since the immune complexes are usually attached to the cell surfaces firmly, the attempt of the neutrophils to phagocytose them goes unsuccessful. Due to this, the neutrophils release their enzymes directly into the tissue spaces over these immune complexes. These are strong lysosomal enzymes that lead to tissue damage, inflammation, and injury to our own cells. This is called phase of inflammation and tissue injury. In SLE, depending upon where these immune complexes get deposited, various manifestations are produced like in blood vessels, it leads to vasculitis. In kidney, it causes glomerulonephritis. In skin, various types of skin rashes. Serum sickness. If the horse got bit by a snake and the snake releases toxins into the blood of the horse, the immune system of the horse recognizes these toxins and sends the antibodies to fight against it, which saves the animal. What we do is we collect the blood of many such animals, which is rich in these antitoxin antibodies. We pool the serum. This was used as antitoxin for snake bites in humans. But drawback is that these types of antitoxin also lead to develop side effects like fever, rashes, muscle pain, joint pain, and headache, etc. What happened is that the antibodies present in the animal serum behaved like antigens for our immune system, which developed antibodies against them. This leads to development of large number of immune complexes in the blood of the patient and got deposited in various tissues and lead to activation of the complement system and neutrophil migration that results in development of symptoms of serum sickness. Type 4. Cell-mediated hypersensitivity. This type 4 is mediated by T cells, the CD4 and CD8 T cells. Type 4 hypersensitivity reactions are of two types. One, delayed hypersensitivity mediated by CD4 T cells. Two, direct cell cytotoxicity mediated by CD8 T cells. Pathogenesis, CD4 T cells. Example, contact dermatitis to plants. This is basically a type of skin allergy to many plants like parthenium, poison ivy. It contains the allergen urushiol. Upon contact with the plant, the allergen gets deposited on the epidermis. Our skin has specialized antigen presenting cells known as dendritic cells, whose main job is to detect such antigens in the skin, capture them, and take them to the T cells. The naive CD4 T cells recognize these antigens and secrete interleukin 2. Now, these naive T cells can mature into two types of T cells. One, Th1 cell, two, Th17 cell. Both of these T cells are different types of T cells that have different functions. If the antigen presenting cell secretes interleukin 12 at the time of T cell activation, it causes the naive T cells to mature into Th1 cell. If the antigen presenting cell secretes interleukin 1, interleukin 6, or interleukin 23, the naive T cells mature into Th17 cell. These mature T cells form an immunological memory of the antigens that lead to their activation. The memory cell stays in the circulation, and upon the re-exposure to the allergen, these memory T cells quickly recognize these antigens displayed by the antigen presenting cells and respond. The Th1 cells secrete many cytokines, mainly the interferon gamma. Here, the interferon gamma acts on the macrophages and alters them in many ways. The ability of the macrophages to phagocytose and kill microorganisms get greatly enhanced. They express more MHC class I molecules on their surface, and they secrete increased amount of interleukin 1 and TNF alpha, which further increases the inflammation. The Th17 cells mainly secretes interleukin 17 and interleukin 22, and their main job is to bring in more inflammatory cells like neutrophils and monocytes to the site of allergy. So, as a result of all this, there occurs infiltration of large number of inflammatory cells in the dermis as well as in the epidermis, which are activated by various cytokines secreted from T cells. These cells try to destroy the allergen by releasing various enzymes by phagocytosing the allergens. And the enzymes released by these inflammatory cells also damage our own skin cells present in the epidermis and dermis. This leads to local cell damage, inflammation, and edema at the site of allergy, which manifests as redness, increased temperature, itching, and development of vascular eruption at the site of contact. Granulomatous diseases. It includes diseases like tuberculosis, sarcoidosis, and leprosy. In these type of diseases, the macrophages cannot remove the target antigen completely. Like in tuberculosis, the walls of Mycobacterium tuberculosis contains mycolic acid which the macrophages cannot break. The macrophages phagocytose these bacteria, but they cannot kill them. Due to this, the macrophages use their next trick, that is, they surround these bacteria and they try to isolate from the rest of the surroundings. The macrophages get morphologically transformed into epithelium-like cells known as epithelioid cells. These cells surround the bacteria and lead to the formation of granuloma, and this gets surrounded by a collar of lymphocytes, and this is known as granulomatous inflammation. Pathogenesis. CD8 T cells. In these reactions, the CD8 T cells do not activate macrophages or bring in more neutrophils, but rather they act themselves and destroy the antigen-bearing cells. Suppose, if we get a viral infection in our body and the viral particles enter our cells and infect them. Due to this, our own cells express certain type of antigens on the cell membranes that basically signals that these cells have been infected with these viruses. The antigens are easily recognized by CD8 T cells, which leads to their activation. The CD8 T cells release perforins, which are chemicals which makes holes in the cell membrane, and after which another chemical known as granzyme is also produced by the same T cells. The granzymes enters the virally infected cells and activate apoptosis in those cells, which is automated cell death. This leads to cell death and removal of these virally infected cells. Similar process occurs in tumor cells, which express certain antigens which are recognized by CD8 T cells. This is followed by killing of these tumor cells by the CD8 T lymphocytes. Type 5 Stimulatory hypersensitivity. It is caused by the interaction of antibodies with cell surface antigen leading to stimulation of cells. In type 2 hypersensitivity, also the interaction between the antibodies and cell surface antigens occur. But there, instead of stimulation, destruction of cells occur. Example, thyrotoxicosis or Graves' disease. It is a disease condition owing to overactivity of thyroid gland. It is caused by an antibody called long-acting thyroid stimulator LATS. It is an IgG type. It acts on the thyroid cell surface antigen, which is basically a receptor for thyroid stimulating hormone produced by the pituitary gland. This causes the release of thyroxine in higher dose from the thyroid cells. This causes the Graves' disease.